Class, welcome to Chapter 10, Part 2 of Biochemistry. This is the last lecture I'm going to have in Chapter 10, so if it's a little bit long, I apologize. If you have not watched and mastered Chapter 10, Part 1 lecture video, I suggest you go back and look at that before proceeding on to this part of Chapter 10. We're going to delve into more detail regarding each of the complexes in ATP synthase, all involved in the process of oxidative phosphorylation. Here's some helpful links. Of course, I want you to read Chapter 10 in your textbook, but I also realize that sometimes the textbook has a lot more information other than what I'm going to cover. And so in that case, I suggest all these YouTube videos, particularly this one. This one gives a great animation of what is going on with the electron transport chain and ATP synthase involved in ATP synthesis, but all of these are really good. I do want to let you know, though, sometimes the nomenclature of complexes 1, 2, 3, and 4 can change from video to video, so whatever I present in today's slideshow is what I found to be the most normal way of thinking about the electron transport chain, and thus what I want you to know for this class. So without further ado, let's get started. So the electron transport chain, oftentimes abbreviated as ETC, is the first part of oxidative phosphorylation. And if you remember, you start with NADH or FADH2, both of which can enter into the electron transport chain. And the overall goal of this is to produce a lot of ATP using chemistry and physics, be it electrochemical gradient or chemiosmosis or electron affinities releasing energy, so on and so forth. Here on the left-hand side of your viewing screen, you'll see a picture of a dam. And the reason I did this is because a dam is a great way to think about what's going on with the electron transport chain and chemiosmosis. So over here, on this part of the dam that I'm indicating with the red squiggly lines is the high proton concentration gradient. And what naturally wants to happen in a dam is that that water is built up, and so it represents a lot of potential energy, and it wants to be let out through some channel in a regulated fashion in order to produce water movement and regulate the balance of water level. And so what you might say is this is low concentration, you might say, or low quantity of water, but it's also, in terms of the analogy I'm using, a low concentration of hydrogen ions. And so what does the pumping? This is going to be your ATP synthase. And what's happening in ATP synthase is all this energy, as that water flows down in my analogy, aka protons flow down through ATP synthase, it harnesses the energy to use for something productive. Okay, in this case, it's going to make ATP by the reaction of ADP plus an inorganic phosphate, PI. Okay, so on the right-hand side, I have an overview of oxidative phosphorylation. And what you have here is complex 1, complex 2, complex 3, and complex 4. And then this is the intermembrane space. So off over here, you have your ATP synthase molecule embedded here that protons can come through and generate ATP by the reaction I previously discussed. What we're going to do is we're going to talk about what each of these complexes are, 1, 2, 3, and 4, the reactions they catalyze, how much protons are being pumped, and we're going to go into a little bit more detail than maybe you've ever been. Of course, there's a whole lot of detail I can go through, and it could take a whole semester just talking about what I'm going over in one lecture today, but we are going to up the ante a little bit and increase the detail of what you may previously already know. Now, what I want to mention about this is that we are going to unpack complex 1 and 2. We're going to talk about what this UQ is. Complex 3, we're going to talk about what cytochrome C is, and complex 4. And we're also going to talk about the final ATP synthesis using ATP. This is the reason we breathe oxygen, okay? It is to derive energy. Oxygen serves as the final electron acceptor in complex 4 in the electron transport chain. And I also want to encourage you to know that if you stop the electron transport chain, you stop ATP synthesis. You run out of energy, and all these protons that are stuck in this intermembrane space, this potential energy will end up being converted to heat energy, and your body will overheat, and that causes a lot of issues as well. And so making sure that this is a well-oiled machine and running as intended is essential for bodily function and normal metabolism. Each of these complexes that we're going to be discussing, 1, 2, 3, and 4, I'd mentioned in the previous video lecture that these are all oxidation reduction reactions happening, which makes each of these complexes, oxidoreductase enzymes. Okay. So hopefully that makes sense to you. Each one's involved in redox. They're all enzymes, or rather they have a lot of cofactors and whatnot, so they're called complexes. So each of these complexes are oxidoreductase enzymes. So let's get started and learn what complex 1 is. Complex 1's name is NADH dehydrogenase complex. And essentially it looks like an L. You may think of this as your phospholipid or your inner membrane. So recall that the mitochondria has a double phospholipid bilayer. It has an inner membrane and an outer membrane. We're talking about the inner membrane of the mitochondria, and this would be the matrix side, and this would be the intermembrane space, okay, the IMS. Okay, so what you see here is you see this part of the L, so it's an L-shaped complex, right? And right here is where I'm talking about. That's the matrix side of this particular complex, and it is hydrophilic, okay? It likes water. It does not associate with the phospholipid bilayer right here, where you have your fatty acid tails coming off. But this part of it does associate it, so we'll call this hydrophilic, and hydrophobic. Hydrophobic, it's scared of water, so it's going to remain in the membrane, so it's associated right here. Okay, now in this complex, what we're going to learn is that, let me delete some of this to save some room. This side of the L that associates with the matrix, this is going to be important for NADH binding. It's going to be involved in FMN binding. It's also going to be involved in the UQ or Q binding site. Okay. And it's going to also have FES clusters. Those are called iron sulfur clusters. And then the membrane domain right here, the one that's the hydrophobic part, this is the membrane domain. It has the proton translocating channels. So it's here in the membrane that the protons actually get pumped. Okay, now let's take a little bit deeper look at what's going on. This is the largest protein component in the inner membrane. So it's larger than complex. I don't know how to spell. Complex is 2, 3, and 4. So it's the largest one. It's L-shaped. So if we're looking at the matrix, as I previously mentioned, it kind of has an L shape about it. It has a hydrophobic region right here, caught up in the membrane, and a hydrophilic region that likes water. It's going to be caught up in the matrix. And the overall reaction that's going to be seen here is you take NADH, and subsequently its electrons get taken from it, and it travels through this entire complex until those electrons wind up on Q. We'll talk a little bit more about Q in a second. Sometimes it's abbreviated UQ. Okay, it's called ubiquinone or ubiquinone. Some people call it different things. And essentially, here's what's going on in this particular complex. I'm going to save some room right here and redraw it. So here's your complex right here. And what happens is, let me delete that right there. You have NADH, which is a byproduct of glycolysis. Remember, it can get shuttled into the mitochondria using the malate aspartate shuttle I briefly mentioned in the last lecture, or it's a byproduct of the citric acid cycle. And what happens is it binds to this particular complex. Sorry, I accidentally slid my finger across the slide, and it makes you have to either redo the slide, so I decided to just recopy the slide, which serves good because I wanted to show you exactly what's going on with this L-shaped complex 1, this NADH dehydrogenase. So I had previously mentioned that NADH comes here to form NAD+, and you may think of it as, and you also get a proton out of it, and those electrons that came from the hydride right here get added here. Okay, now what really happens to those? Here's what we're going to show. I told you in the previous slide that there's an FMN binding site. So what happens is there's an FMN. It's a flavonoid, and what happens is whenever it accepts those two electrons, it becomes FMN. Oh, I love the glitches, y'all. FMNH2. Now, where did those two H's come from? Well, two protons from the mitochondrial matrix can join with it, with those two electrons from NADH to form FMNH2. Now, those two protons then leave, and then those two electrons can then get transferred to what we call iron sulfur clusters. Okay, there's a series of them. There's a different number per eukaryotic cell, and so we're not going to get too much into it, but just think of these random iron sulfur clusters, which they're not random at all, but they pass electrons from one to the other. And they can, and what's neat about these is that it starts with low electron affinity, and what drives this is that by the end of your electron passing from the first iron sulfur to the final one, it slowly gains higher affinity for electrons. And so you have a low affinity to begin with, and then the reason they keep on passing from one to another is because the next one wants electrons more than the previous one. And also they're in close enough proximity for an electron jump to happen. If they were too far away, the electron would not be able to make the jump. And so they're really close together, and they increase affinity going that direction. Okay. Now, the electron's getting passed around this complex, until it eventually finds its way. It's still going through the iron sulfur clusters. And then you have Q right here. Q is coenzyme Q. As I said, we're going to talk a little bit more about that in just a little bit. But Q is called coenzyme Q. It's also called ubiquinone. And what happens here is that those two electrons get transferred to this Q, and then two protons also get transferred to this Q. And what happens is whenever that Q combines with two protons and two electrons, it becomes QH2. QH2 is a transporter molecule. It essentially just transports electrons across the membrane. Not across the membrane, but from one complex to another, excuse me. And that's what's going on in complex 1. Now, I failed to mention all the protons being pumped. Now, whenever you are doing your iron sulfur cluster electron passing, what happens is whenever electrons get passed from one to another, there's a little bit of energy that is released. Okay, so as you go from this iron cluster to that iron cluster to that iron cluster, you can think of energy being released. And what happens is that energy is high enough to where protons over here in the matrix are being passed across to the intermembrane space, only because the iron sulfur clusters are passing around an electron. And so what happens at the end of complex 1 is you just pumped 4 protons to the intermembrane space. So what happened at the end of complex 1? You pumped 4 protons into the intermembrane space. Remember that I said in the previous slide that the arm that is associated with the phospholipid bilayer, aka the hydrophobic part, class, I need to learn how to talk. The hydrophobic part is called the proton translocating channels. And do you see that? They translocated protons from the matrix side to the intermembrane space, thus increasing the proton gradient. And so you need to know this. Complex 1 gives off 4 protons to the intermembrane space. FMN goes to FMNH2, so FMN is the first electron acceptor in this complex. It then passes the electrons onto the iron-sulfur clusters. Low affinity going to high affinity. They're close enough to where an electron can jump, which causes an energy buildup, which then allows protons to make the jump to add 4. And so that's what's going on with complex 1. Let's take a look at what these iron-sulfur clusters are now. Here's an image right here of the iron-sulfur clusters that are seen in this complex, or actually all the complexes. So if you look, here's your iron right here, and it forms a sort of lattice structure right there with sulfur molecules. And notice that they are bound to the protein part of this complex 1, this NADH dehydrogenase, which, by the way, that name hopefully makes sense because you dehydrogenated, you took a hydrogen off of NADH to start off this whole complex. So NADH dehydrogenase. You dehydrogenated NADH. Now, after FMNH2, it passes the electrons through these iron-sulfur clusters. And notice the protein component of this NADH dehydrogenase has various cysteines located in very strategic regions. So the cysteine residue, recall, has sulfur in it, which can form disulfide bonds. But not only disulfide bonds, look, that it can also form a bond between the sulfur of the cysteine and the iron of the iron-sulfur clusters. These iron-sulfur clusters, as you can see, always from a cysteine residue of the protein part, and then the sulfur residue, the sulfur part of the residue is bound to the iron. These have a certain affinity for electrons. And as I said, they differ in structure. See how one looks like a cube and the other looks like a square. And what I want you to know is, like I said in the previous slide, they are increasing in affinity as you pass the electrons from the beginning of the complex to the end of the complex. And what that creates is every time you jump, make the electron jump, it creates an energy that can be used to translocate those protons to the other side of the membrane. And recall that these series of iron-sulfur clusters are part let me see what I'm trying to draw here. So here's your membrane, here's your matrix side. Like where the citric acid cycle is. These iron-sulfur clusters are in this region, okay? So they are in the hydrophilic region. They like water. The only thing that's in this region is the proton translocating channels that take the protons from this side, which are in low concentration already, and put them on the other side. So you're kind of filling up the dam on the high potential energy side of it. Is there anything else I wanted to say about this? All right, well, it seems like that's all I wanted to say about the iron-sulfur clusters. So that's what they look like, and they pass electrons throughout these complexes. You're also going to see these iron-sulfur clusters in other complexes. All right, ubiquinone. You heard me say that this is called coenzyme Q. Coenzyme Q. It's called ubiquinone, like I said. Sometimes it's abbreviated as Q or UQ. Yeah, Q or UQ, you'll see. I'm going to use Q in my slides. It is lipid soluble, which means that if you look over here in your drawing, in the drawing I provided, there's UQ right there. Do you see how it's associated with the hydrophobic part of the membrane, meaning it's in the fatty acid tails of the phospholipid bilayer? And so it's not associated on either side of the membrane, but rather it's inside the membrane itself. So it travels through the little the little tails of the fatty acids. It's not stationary. It's mobile. It moves from one to the other, one to the other. And what it does is, you see complex 1 right here. It takes one electron at a time And so what happens is it becomes, it's ubiquinone here. And then after it accepts one electron, it's semi-ubiquinone. And then after it accepts two electrons, meaning that one and that one, it's called dihydroubiquinone. Dihydroubiquinone. And what happens is, we're going to talk a little bit more about this in a second, but it's involved in two ways, okay? So once the ubiquinone takes the electrons from complex 1, one at a time, ubiquinone to semi-ubiquinone to dihydroubiquinone, sorry if I didn't spell that well, dihydroubiquinone, only whenever it has those two electrons is it going to be mobile, okay? It's going to be a transport protein, and what it's transporting is electrons. Now, the electrons from complex 1 do not go to complex 2, and I can't stress that enough. They go straight to complex 3. The electrons from ubiquinone do not go to complex 2. It skips complex 2. Now, what you're going to see is that here's complex 1, and then the Q here will go straight to complex 3. But then you also have this complex 2, which we're going to talk about. It also has a Q associated with it, and this one's associated with FADH2. The electrons from that can also go to complex 3. So what's happening here is the electrons from 1 get passed to Q, and Q goes straight to 3. If FADH2 gives electrons into complex 2, those electrons wind up on Q, and then those electrons from Q go to complex 3. So complex 2 never goes to 1, complex 1 never goes to 2. Both of them use Q to go to complex 3. Okay, so I just wanted to be clear about that. And it does transport 2 electrons at a time. They just bind to it one at a time. So one binds semi-ubiquinone, the second one binds dihydroubiquinone, and then once 2 are on it, it gets shuttled across the membrane as the reduced form, QH2. So Q, it gets 2 electrons, and it also takes 2 protons, and it becomes QH2. Whenever it's QH2, that's when it gets shuttled to complex 3. Complex 3 receives all that, and then it gets oxidized back to Q, and then goes back to where it's needed, okay? Okay, so you reduced, that's the reduced form. This is the oxidized form. And like I said, once it loses the 2 H's and the 2 electrons at complex 3, it goes back to where it's needed, be it complex 2 or complex 3. So now let's get on to complex 2. Complex 2, you've already seen before. It's called the succinate dehydrogenase complex. Now, this is the same exact enzyme or complex seen in the citric acid cycle. You might have heard me call it the succinic dehydrogenase. More commonly, it's actually called succinate dehydrogenase. This is the exact same process seen in the citric acid cycle. So what you see here is actually a step in the citric acid cycle is the same exact thing going on in the electron transport chain. Okay? This is not a proton pump, but it certainly does take electrons and shuttles them to complex 3, which then serves as a proton pump. So indirectly, it does pump protons, but not in itself. So complex 2 does not give any protons to the intermembrane space. The reaction is going to be succinate to fumarate. Sound familiar? Citric acid cycle. And what happens is FAD goes to FADH2. Ever wonder where the H2 comes from? Well, it's the low concentration of protons in the matrix that are already there. Okay, so it releases FADH2. And the FAD to FADH2 occurs, so you reduce FAD, and keep in mind, succinate to fumarate is an oxidation. So this is an oxidation reduction reaction. And so also the succinate provides a proton to reduce FAD to FADH2. So this is in the citric acid cycle. Now what happens is, here's what complex 2 looks like, succinate dehydrogenase. Recall I said in the previous slide that the electrons from QH2 do not go to this complex. It goes straight to complex 3. And so here's what happens. I'm trying to think of the best way to present this. I think here is a good way. So let's say that you have your membrane and you have, trying to imitate this drawing right here. So this is complex 2. This is matrix. This is intermembrane space. Now, what happens is you have succinate go into fumarate, and in this process, FAD I've always wondered why it does this. This online teaching is going to drive me insane. FADH2. And then FADH2, then whenever it gives protons back here to the matrix side, dishes off 2 electrons. You see that? To be oxidized back down to FAD to complete this process. And so here's why FADH2 gives off energy, because it donates some electrons to the succinate dehydrogenase complex, which is complex 2 in oxidative phosphorylation. These electrons then get passed through, you know it, FeS clusters again. And again, the gradient, the potential energy, and all this. But the only difference is this one does not pump protons, okay? So it just passes through the iron sulfur clusters until, as I already previously mentioned, they get bound to Q, coenzyme Q or ubiquinone. And the same thing happens. You bring in those 2 electrons, you bring in 2 protons from the matrix, and it goes to QH2, which then gets shuttled onto complex 3. Okay, so this one acts a lot like the NADH dehydrogenase. You have a molecule that binds to it, and so NADH in the first one, FAD on this one, and FAD gets converted to FADH2, which then donates its electrons, just like the electrons from NADH are donated to the NADH dehydrogenase. Those electrons get passed through iron sulfur clusters. This one, however, does not pump protons. The final acceptor of these electrons would be Q, ubiquinone, aka coenzyme Q or UQ, and then whenever those 2 electrons bind to UQ along with 2 protons, it becomes the reduced form of Q called QH2, which then is mobile, or mobilized rather, to complex 3, where it's going to dish off its electrons to continue the electron transport chain. And yeah, and recall that this Q does not ever go to complex 1. Here's complex 3. Recall that it received all the electrons from Q. That's what these arrows right here are trying to indicate. QH2 from either complex 1 or 2 would be giving its electrons to complex 3 here, as shown. Complex 3 is called the cytochrome BC1 complex. If you see cytochrome, that word, it means that it contains a heme, or multiple hemes. Remember, a heme is an iron-containing porphyrin ring-like structure. You saw the heme and the hemoglobin, right, as we studied that. This complex only has 1 iron-sulfur cluster, and there's not a whole lot to say about this, at least as it pertains to this lecture. I just wanted to say that electrons pass through it, really. Let me see. I'm getting lost in my notes. I didn't staple my page correctly. So the overall reaction, 2 protons. Sorry. You have UQH2, which is also I called QH2, which eventually go to cytochrome C. Cytochrome C right here. And the net effect you have 4 more protons pumped into intermembrane space. Pumpered, right? Pumped into intermembrane space. And you also have 2 electrons that are fed off into intermembrane space. Now, that sounds weird, right? Intermembrane space side to cytochrome C. Now, notice in the drawing right here, cytochrome C, so this is your matrix side. So the cytochrome C is in the intermembrane space. And so you have to keep track of where things are. And so cytochrome C is associated with the intermembrane space side, or you can think of it as the external side of the inner membrane. So it's on the side where all the protons are, the proton gradient, the high concentration. And so remember, Q was in the hydrophobic region, and this one's in the intermembrane space side region. Now, what happens here is cytochrome C is a mobile electron carrier. It's mobile, so it can move. It's not stationary. And this time, it only transports 1 electron at a time. So that's called cytochrome C. You get 1 electron bound, then it goes over here to complex 4, and then it goes back, and then another electron is added because that one is now given off. 1 more electron binds, and then it repeats the process. Does that make sense? So it's just a ball rolling back and forth, donating 1 electron at a time. So never does it accept 2 electrons. Okay? It never accepts 2 electrons. And what happens as a net effect is, like I said, 4 electrons are pumped into intermembrane space. Okay? Where do those 4 protons come from? 2 from QH2. So 2 from the reduced form of the cytochrome C. And the other 2 come from the matrix pool. So remember, you have your membrane. That would be called the matrix. This is the intermembrane space. And then you have a low concentration of H+. And so in complex 3, 2 of those come over. Okay, and the other 2 come from QH2 to then reoxidize it after those hydrogens or those protons are gathered from it. Okay, and essentially all what's going on in complex 3 is electrons are passed right through it. Okay, so this is complex 3. You have QH2. That's the matrix. It goes to Q. Those 2 protons are pumped over. Sorry, I'm just going to go ahead and finish my drawing. This is coming from complex 3. I said that QH2 come over. That was a glitch. I accidentally hit the screen wrong. And then those 2 protons come over. 2 H pluses come from the matrix. And then the electrons travel through the complex to find, I guess I should put it here, cytochrome C, where that goes to complex 4, transporting 1 electron at a time. Okay, so hopefully that made sense. That actually goes with the previous slide, so sorry for hitting the button. I've just lectured on that slide for 10 minutes or so, and I didn't want to have to restate all that. Okay, complex 4. Here's our final complex of the electron transport chain. This is the cytochrome oxidase complex. Cytochrome oxidase complex. This one's a little tricky. So it catalyzes 4 electron reduction of oxygen to form water. O2 can only accept 1 electron at a time. And this one does not leak electrons out like other complexes. Remember the other complexes, it either gives it to Q or it might give it to cytochrome C. This one finishes off, you know this from Dr. Ladine's class, it finishes at oxygen as the final electron acceptor. Now, whenever you take, the 2 electrons originally derived from NADH, what happens is you get this reaction 1/2 O2 plus 2 protons goes to water. But this complex 4, is actually only complete whenever you have 4 electrons, aka cytochrome C goes 4 times over because it transports 1 electron at a time. So the real reaction is O2 plus 4 protons goes to 2 H2O. So this complex is fully active when 4 electrons are present, and so that is a balanced chemical equation. What happens in this case when you only deal with 2 electrons is you get 2 protons pumped. But whenever you actually take into account all 4 electrons that really make this entire complex active, you can think of it as 4 protons being pumped, twice the amount of what I just said. That's a little bit confusing. All what I'm trying to say is this is not a full chemical reaction. Have you ever seen 1/2 of an O2 react? But with 2 electrons, this is the equation. So that's what I'm saying. You need 4 electrons to actually get a full oxygen molecule to react to give you that reaction, which gives you 4 protons into the intermembrane space. Okay? 4 protons to the intermembrane space. So this is why you breathe. This is why you need to maintain good oxygen levels in your body. So here's oxygen in the hydrophobic region. And what happens was the electrons that came from cytochrome C right here, after it brings over 4 electrons, it travels through these copper centers and then iron copper centers, different subunits, more copper centers, a subunit right here, until it gives you 4 protons from your substrate. 4 protons from your substrate. That's just in the space, okay? That's just in the space. 4 protons that are pumped from the ATP synthase, and then so you have your protons, you have your oxygen, and then you have your 4 electrons from your cytochrome C, giving you your 2 waters right here. So electrons coming from cytochrome C 4 times over because it transports 1 at a time. Oxygen from breathing, and then protons from the intermembrane space. Sorry, the matrix. The matrix side, all combined to give you water. So oxygen is your final electron acceptor. Okay, now let me see. It's composed of 2 monomers, 2 monomers with 14 subunits. So it's really big. Go back to your protein chapter to see what that's all about if you don't understand. And what it has is it has hemes. You knew that because it's called cytochrome oxidase complex. Oxidase meaning it's an oxidoreductase kind of enzyme. Cytochrome meaning that it has hemes. It has iron copper centers. And it also has copper ions. So there's a lot going on, but just know that in a full net reaction, it dishes out 4 protons to the intermembrane space, thus increasing that proton gradient. And it also, but if you were to only consider 2 electrons, because what I'm saying is that it takes 2 NADHs to get 4 electrons, or you can say 2 FADH2s or a mixture of the 2. But in order for this complex to be fully active, giving you that full chemical reaction, giving you those 2 waters, it's going to pump out 4 protons and take 4 electrons. Okay. And here's the last part of our electron transport chain, ATP synthase, the chemiosmosis part. This is such a fascinating enzyme, and I love talking about it. I'm not sure how much depth I'm going to go into it in this particular slideshow or any in this particular class, because there's just so much to say about the ATP synthase. But recall that chemiosmosis is the process in which you have high proton concentration and you have a low proton concentration. And what happens is these up here are full of potential energy. Why? Because there's a bunch of positive charges bumping into each other because they're in close proximity and they want to repel each other. But also there's a osmosis gradient, an electrochemical gradient, in which the high concentration wants to flow to low. But remember that this bilayer, this membrane, is not permeable. And so they can't get to where they want to go, which causes a lot of potential energy buildup, just like water stuck behind a dam. And so the molecule that helps accomplish this is called the ATP synthase, which kind of looks like this. I'm drawing it upside down from the drawing over there. And it kind of has this side arm right here that connects the two sides of it. And down here, that's going to be called the F1 unit. And up here, it's called the F0 unit. F0, F1. The F0 is called the transmembrane channel. The transmembrane channel. And the F1 unit is called the ATP synthase. So we're looking here. And we're looking here, transmembrane, because it spans the membrane. Hopefully you can see that. You can see that that's F0, that's F1. Notice here that the ATP synthase part, they're called alpha, beta, alpha, beta, alpha, beta. There's 6 of them. It actually rotates like a motor. So it's rotary action. And what else do I want to say? Oh, this is called the flexible stator. Well, it is flexible. That describes it, but it's called the stator. So you can see it there. The stator. So the F0, the the part that's the transmembrane channel, the F0 motor converts the proton motive force, because recall that it is touching the high concentration gradient up here. High concentration gradient, so it's close to it. It converts the proton motive force into a rotational force of the central shaft that in turn drives ATP synthesis. And so as it turns, remember ADP plus PI get converted to ATP. As it turns out, 3 protons entering into this stator gives 1 ATP, because remember the energy derived from the proton gradient and ATP synthesis moving the rotor, it's that energy that even drives ATP synthesis. And as it turns out, once you have 3 protons enter the molecule, the ATP synthase enzyme, it finally gets enough energy to throw a phosphate on that ADP to generate ATP. Okay, and I'm trying to think. You can look at some of the animation and drawings if you want to see what's going on here. There's a lot of details I can go into. There's, for instance, an L site, a T conformation, an O site that you can read about in your book, and then the cycle repeats. It's a little bit confusing, but what I do want you to know is what I've presented in this slide. You have an F0 and an F1 unit. The F0 is the transmembrane channel hosted in the phospholipid bilayer. The F1 part, called the ATP synthase, is on the matrix side. And it's the ATP synthase region that actually binds your ADP and your phosphate and gives you ATP. And then it leaves and floats around in the matrix to be used wherever it might need to be used at. So hopefully that makes sense. Take a look at some of them videos at the beginning of this slideshow to get further visualization of what's going on with this fascinating enzyme. In summary, this is what's going on with the electron transport chain. So notice here is the matrix side. Out here is the intermembrane space. And so you have complex 1, the sodium dehydrogenase complex, which shuttles four protons into the intermembrane space. Complex 2, succinate dehydrogenase, that's involved with FADH2. Succinate goes to fumarate. Those 2 electrons from FADH2 get transported to Q or UQ. Same thing with complex 1. That molecule located in the phospholipid bilayer part, the fatty acid side, the hydrophobic portion, gets it transports those electrons to complex 3, which then is able to shuttle 4 more protons to the intermembrane space. And it passes those electrons on. And some of those protons came from the QH2, if you remember. And then it passes its electrons to cytochrome C, which passes 1 electron at a time to complex 4. The reaction gives 2 protons when you consider using 1/2 O2, but really the complex is fully active whenever you use, whenever you use 4 electrons to generate 4 protons in the intermembrane space, giving you, instead of a half number there, a full O2 plus 4H pluses gives you 2H2O. ATP synthase then takes the water from built up here like a dam, all this potential energy, high concentration, sends it down here. The potential energy is then taken to spin a rotor in the ATP synthase, and that energy derived by 3 protons gives you 1 ATP molecule, which is used for energy. That proton then finds its way out of the ATP synthase to be in the low concentration side of the mitochondrial matrix, to be used in this side to then be pumped over again using some other process in one of the complexes. Wow, that's a lot of stuff. So hopefully you got a lot out of this. Hope that I actually was able to teach you something that you didn't already know in your other classes. Let's go on to the conclusion slide now. Study this material over and over again, class, until you master this material. Send me emails, watch videos, build a strong foundation. I've been told time and time again from my pre-med students that biochemistry is a heavy portion of the MCAT, so you do want to have good command of this material. I encourage you to read the chapter and also to remember Romans 1:25. They exchanged the truth about God for a lie and worshipped and served created things rather than the Creator, who is forever praised. Amen. Emphasizing that God created this. I can't even write well. God created all this. And sometimes as scientists, we think that we have command over nature. We can govern it. We figured it out. Look how cool we are. We can even tell you that we don't need a God to describe nature, but just take a step back and think about what all we talked about here in chapter 10 as it couples with also chapter 9 and the citric acid cycle and glycolysis in chapter 8, and how gluconeogenesis and glycogen synthesis and degradation is all regulated. We have a great God that created this complex system, this complex biology that we live life with, and it's simply governed by physics and chemistry. Recall that these chemicals don't have brains. They're not thinking. They're simply acting upon the nature of chemistry and physics to achieve life. So worship and serve the Creator today, and just think about what God did and praise the Creator for what He's done. Hope you all have a blessed day. Hope you enjoyed this lecture.